Hello, everyone. I appreciate the opportunity to be with you today. I am here to talk to you about CloudClone Corp. And its main business. I will introduce CloudClone, core products, CRO service, and customer service, respectively. CloudClone Corp. is a high-tech enterprise focusing on the research and experimental development of molecular biology and immunology, as well as an original manufacturer of core biological reagent raw materials. Founded in 2009, CloudClone has 14 years of experience in scientific research and 14 years of product accumulation. As we all know, a company must have the ability of independent research and development if it wants to stay in the forefront of the industry for a long time. CloudClone has been adhering to the tradition of independent research and development of core technologies, adhere to original production. So far, we have achieved independent research and development of all products. First of all, let me introduce the laboratory research conditions of CloudClone. Currently, we have more than 10,000 square meters of laboratories in total. The first thing you see is the building of CloudClone main company in Wuhan, with 5,500 square meters of laboratory, mainly used for the production of enzyme-linked immunosorbent assay kit, antibody, protein and other reagents, as well as the research and development of new products. The picture on the top right is CloudClone Animal Inc. in Wuhan, a 6,700 square meter laboratory for animal experiments and CRO services. In addition, based on the fact that more than 75% of our customers are located in European and American countries, CloudClone has an office in the United States, which are mainly responsible for the promotion of brands abroad and the promotion of corporate culture. This office is located in Houston, the United States. Currently, CloudClone's Animal Center has 2 certificates. We have Laboratory Animal Use License and Laboratory Animal Production License issued by Hubei Provincial Science and Technology Department. The total area for raising animals is 1,200 square meters, and the area of SPF Animal Room is 467 square meters. You can see the environment of our animal room from these pictures. The experimental facilities of these animal rooms are mainly to raise some experimental rabbits, rats, and mice. Using these experimental animals, we can complete the preparation of antibodies and animal models of diseases. In the future, we will also prepare transgenic animals here, because we use our own experimental animals, and the products produced on this basis can ensure 100% traceability of the products and ensure the product quality to a certain extent. The products of CloudClone have also been tested by customers. Since the establishment of the company, more than 24,000 SCI papers have been published using CloudClone products, with an annual increase of more than 25%. Because more than 90% of these SCI papers come from our ELISA products, CloudClone was awarded the Best ELISA Kit Supplier Award by CITEB in 2020. Among these SCI papers, there are more than 260 papers published in top journals such as Cell, Nature, Science, etc., including some cover articles, further explaining that our products are recognized by customers. Then I will introduce the core products of CloudClone, which are mainly assay kits, proteins, and antibodies for scientific research. The assay kits of Cloud Clone are divided into traditional ELISA kits, chemiluminescent immunoassay kits, and Luminex kits. For traditional ELISA and chemiluminescent immunoassay kits, Cloud Clone has also developed products with different characteristics according to market needs. First, Common Kit. Cloud Clone has developed more than 8,000 common assay kits after 14 years of accumulation, which can meet the protein and small molecule detection needs of most species in scientific research. Second, for some small experimental animals, such as mice and zebrafish, due to the limited sample size, Mini Kit has been launched, which only needs 25 microliter for detection. This type of kit is based on the high affinity and highly sensitive antibody pairs of Cloud Clone, so the detection results can be guaranteed under the condition of reduced reaction system. Third, Antibody ELISA kit used to detect antibodies, including endogenous autoantibodies and vaccine protective antibodies, used for the detection of autoimmune diseases and some viral infections. Fourth, we also have Instant Kit that is quick and easy to use. The reaction time of the common kit is about 3 hours, while the experimental time of the Instant Kit is 1/4 to 1/3 of the common kit. It is suitable for some outdoor sampling or when sample freshness is required, which can improve the experimental efficiency. Fifth, there are also high-sensitive kits with a detection sensitivity of 1 pgml, which can be used to detect targets with very low content, such as cytokines, by using high-quality monoclonal antibodies with high specificity and efficient signal amplification system to improve the sensitivity of the kit. Finally, we also have a wide-range kit, which can be used in the case that researchers do not know the approximate concentration of the target when conducting research on some new species and new indicators. It covers a wide range and avoids the problem that cannot be able to get precise results, because it is not in the detection range. In addition, based on the latest Luminex XMAP technology, Cloud Clone independently developed Luminex kits. More than 10 targets can be detected using only 20 to 50 microliter samples at a time. Luminex is based on flow technology, which is more accurate than ELISA based on color change. Luminex kit does not have the problems of color change control or fluorescence attenuation that traditional immunoassay kits have. Its detection sensitivity is 10 to 100 times that of common ELISA kit, and the detection width can reach 1,000 to 10,000 times. It is applicable to the situation where the sample size is small and there are multiple targets to be tested. These are highlights products of assay kits. According to the published papers, we collected information on the use of the products, the number of papers by using ELISA kits for detecting cytokines such as TNF-alpha, IL-6 and IL-1B has reached hundreds of levels. These figures are the comparison of the citation amount of some small molecular assay kits retrieved from CiteHub. It can be seen that the citation amount of Cloud Clone's small molecule assay kits, such as testosterone, corticosterone and melatonin, is much higher than that of Company R and Company A. To some extent, it reflects the advantages of Cloud Clone in the small molecule assay kits market. So, how can we guarantee the quality of so many such good assay kits? Because the kit of Cloud Clone is completely developed and produced independently, we can carry out 3-level quality control on the product, including raw material testing, semi-finished product testing and final product testing. Quality control first goes through in-house test, including sensitivity, standard curve, recovery rate and precision analysis, etc. The coefficient of variation within and between batches of the kit is within the standard range to ensure the reliability of the product. In addition to in-house test, third-party companies have also verified the quality of our products, which further guarantees the quality of Cloud Clone products. This is a kit test report issued by them. These are pictures of the assay kit labs and production center. The first is the preparation and screening of paired antibodies. Then through an automatic production equipment chain, to complete the steps including plate coating, plate washing, bottle washing, filling and so on, improve the production efficiency while ensuring the accuracy of products. The final semi-finished products are stored in the warehouse for subsequent inspection and production of finished products. The next introduction is protein product of Cloud Clone. After more than 10 years of scientific research and development, Cloud Clone has 4 major protein production platforms, including multiple expression platform, protein extraction platform, peptide synthesis platform, and small molecular modification platform, corresponding to the production of recombinant protein, native protein, peptide and coupled small molecule respectively. The protein expression system of Cloud Clone can be divided into Escherichia coli prokaryotic expression system, eukaryotic expression systems such as yeast, insect cell and mammalian cell, as well as bioreactors that can be produced in batches. Through these expression systems, Cloud Clone can produce more than 15,900 protein, covering multiple species such as human, rat, mouse, pig, horse, bovine, goat, etc. And we have screened more than 1,000 active proteins including TGF-beta1, TNF-alpha, IL-4, IL-2 protein product. The activity of these active proteins has been verified by the company and customers' targeting activity experiments, and the results show that the proteins have one or more functions. The other proteins are not without activity, but the activity has yet to be verified. In addition, Cloud Clone has GMP-level bioreactor. The production of product reaches G or even kg, can meet the needs of industrial customers. The target end users of Cloud Clone protein products can be roughly divided into 2 categories. Industrial-grade protein product end users are mainly some in vitro diagnostic, healthcare, and cosmetics companies. Our proteins can be used as raw material for medicine, diagnostic reagent, cosmetics, and scientific research reagent. Low-dose protein is mainly for biomedical researchers, medical researchers, universities and research institutions to carry out some basic biomedical research. These are pictures of protein production labs. First, complete the cloning of the protein gene, the construction of the expression carrier, and the protein extraction and purification work in the molecular biology labs. This is cellular culture and the detection of protein expression in the cellular biology labs. These are biological chemistry labs, which are mainly used to complete the work of polypeptide synthesis and protein modification. These purified proteins will perform some quality control detection in the quality control labs. The antibody products of Cloud Clone can be divided into first-generation antibodies, polyclonal antibodies, second-generation antibodies, monoclonal antibodies, and the third-generation antibodies we are now focusing on development, recombinant antibodies. At present, the characteristic antibody products of Cloud Clone include small molecule antibodies which are rare in the market. We have a variety of small molecule antibodies which many other companies have few or even no. It is because Cloud Clone can produce high-quality small molecule antibodies that our small molecule assay kit can occupy an advantage in the market. Then there are stable recombinant antibodies preserved by genes. Compared with the monoclonal antibody produced by hybridoma technology, the recombinant antibody can avoid the loss of antibody caused by contamination or mutation of hybridoma cells. At the same time, it gets rid of the dependence on experimental animals and reduces the difference between batches caused by different animal batches. There is also a labeling antibody that can be linked with different labels, including enzymes, fluorescein, biotin, and flow cytometry antibodies. As with the assay kits, the antibody quality is controlled through in-house test and third-party validation. These in-house tests include conventional Western blot and immunohistochemistry. All antibodies have been tested. Some antibodies conduct immunofluorescence and flow cytometry as needed. In addition, we also carry out knockout verification on the antibody to prove the specificity of the antibody. Cloud Clone is a supplier of raw materials for biological reagents. Third-party companies will also test our antibodies when purchasing our antibody raw materials. These are their immunohistochemistry and immunofluorescence verification data, which prove the quality of Cloud Clone's product. These are pictures of antibody production labs, including the polyclonal antibody labs, the monoclonal antibody labs, and the antibody quality control labs. These pictures are the immunohistostainer, digital scanners, and flow cytometer that we introduced to complete some related experiments. Furthermore, Cloud Clone can also provide CRO services. The first is primary cells, which are widely used in the field of basic research in molecular, cell biology and biomedicine, and can also be used in biomedical industries such as drug screening, drug metabolism and toxicology research. At present, Cloud Clone can provide more than 1,000 primary cells of multiple species, which can meet most user needs in the market. Therefore, primary cells are widely used and have broad market prospects. And then animal disease models of Cloud Clone, which are mainly used for experimental physiology, pathology, and therapy research for human diseases. Because the process of using people as subjects to delve deeper into the mechanisms of disease is slow, and many experiments are ethically limited. These problems can be well avoided by using animal models. At present, Cloud Clone can provide more than 800 animal disease models in multiple species, including cardiovascular and cerebrovascular diseases, respiratory diseases, digestive system diseases, etc., and comprehensively cover human common disease research. This is the entire CRO service process. According to the needs of customer projects, we can complete the entire process from information collection, experimental design, feasibility assessment, experimental operation, and data analysis. The entire experimental operation is real-time video for customer acceptance. Moreover, based on the assay kit, protein, antibody products, and animal experimental facilities owned by CloudClone, we can enable customers to enjoy one-stop services and improve service quality and service efficiency. We also set up a monument to experimental animals in memory of those who sacrificed their lives for human health. Finally, CloudClone provides complete pre-sale and after-sales service. We guarantee that if our products and services do not give customer a satisfactory experience, a full refund, credit or replacement could be offered. In addition, during the entire service process, we will provide full technical support services by professional master and doctoral team and promptly reply within 24 hours. That's all for my introduction. Thanks for your attention.